students of Standard 12. Today we are going to end the chapter on mutation. So a little bit is left, so we will like to study that part. As I told you, mutations are of two types. One is called induced mutation, another is a spontaneous mutation. I just try to recollect that the induced mutation is something where it is a manipulated, handiwork of human being, human interference is there in the gene, so that there is a change which can be harmful, which can be useful. But generally harmful genes are not induced, but sometimes it can come out. Okay, like say people are telling about the coronavirus, maybe it may be, it is not sure, that may be a handiwork of human being, human interference, and that may have created a mutated gene. But induced mutation can also be used for various purposes. Like for example in agriculture, a lot of mutations are done, polyploidy are done among various types of, like say for grasses, in wheat and all these things, for creating special varieties. Spontaneous mutations occur in the nature. Now this one is usually like say the sun rays, the electromagnetic waves, the cosmic rays. These also are a type of mutagens which can induce mutation in various types of crops naturally, and new varieties of crops can arise. So spontaneous mutation lead to evolution, which we call a natural organic evolution. So they do help in this. Now as we know that these mutations are done by various types of agents. Now these are called mutagens. You can see here, like there are certain physical mutagens, like say for ultraviolet rays, which is reaching to the Earth's surface, after crossing the barrier of the ozone layer because of the ozone hole, and then this can mutate genes in plants and animals. So exposure to too much of sunlight for human being is also not good, because that can cause certain change in the gene. Gamma rays are there, beta rays are there, alpha rays are there, which are radioactive rays. They are also, like beta rays are generally electrons, so we do not consider them so much harmful. But gamma rays and alpha rays, these two rays in the radioactive particles, which are given out by radioactive particles, they are physical mutagens. Chemical mutagens are there, like say nitrous acid is a type of chemical mutagen, 5-bromouracil is a chemical mutagen, phenol is a chemical mutagen, bromine are chemical mutagens. Now even colchicine also is a type of chemical mutagens. Now these chemical mutagens, what do they do? That sometimes they pair a different pair which is a different pair than which is the normal pair to a certain basis. Now, so that creates a mutation in the gene. So these are the different types of what we call physical and chemical mutagens. Now we have already seen this. So that is all about. Now what do the mutations actually? How do they help us? Like mostly, mutant genes are deleterious, or they may not be useful. They may be lethal. But in many cases, mutation is done purposely to create certain varieties in the plants and animals. Like say for example, you can induce a mutation even when we introduce a gene into another gene. That is also, we are creating a mutant gene. Like say for example polyploidy, which is naturally induced, that is a type of mutation. Like say for example the hexaploid wheat or the tetraploid wheats that have been generated by using colchicine. Colchicine is a type of chemical which arrests the formation of the spindle fiber during mitosis. Now when the chromosome is dividing, suppose there are 16 chromosomes in a plant, and then 16 is dividing into two parts, 16 and 16. In between, if you add colchicine, then what happens? The spindle fiber arrested, the doubling of the chromosome number takes place, which is an unusual event. Now that is a type of mutation. So you can double or triple, so you can produce triploids, you can produce tetraploid, you can produce hexaploid, like this type of polyploids are done. And there are different types of polyploids, like autopolyploids or then allopolyploids are there. If you have same type of chromosome doubled, then we call it autopolyploids. If there are two different species of plant or animals and there you double the gene, then it is called an allopolyploid. Now this is all about the chapter on mutation. We are going to start a new chapter today, that is the chapter on reproduction in organisms. Now this in earlier classes we have studied about this, but we are going to study a little bit in more detail. Like say for example, what is reproduction? Producing identical species from a parent plant or animal is called reproduction. Now there are two types of reproduction. One is asexual reproduction, another is sexual reproduction. Now what is asexual reproduction? When we produce an individual from any other part than either other than the reproductive part, where there is no meeting of the gamete and there is identity of the gene among the parent and the offspring, then we would call that asexual reproduction. In sexual reproduction, of course, there is two types of gametes from the parent, like the male gamete and the female gamete. They fuse to form the new individual. So there will be, of course, variation. They will not be identical to the parent. Now, what is the lifespan? Lifespan is at the birth to the natural death of an organism. Every organism is born, then it carries on and completes a life cycle, and then has a natural death. That is called the lifespan. Now, lifespan of a banyan tree is 200 to 250 years. You can see lifespan can be very high, it can be very short also. Like a parrot has a very large lifespan of 140 years. A tortoise has a lifespan of about 150 to 200 years. So, the lifespan varies, but there are many organisms are there, maybe few minutes or few hours they survive. So, that lifespan varies in every organism and individuals. Now, in asexual reproduction, what do they produce? As I told you that they are genetically identical to the parents. So naturally, they are producing clone. When we produce through asexual reproduction, we are producing the clone of an individual. They will be genetically alike to the parent from where it is derived. Now, the fission is asexual reproduction are various types. Like first is fission. Fissions are two types. One is called binary fission, another is called multiple fission. In binary fission, usually the cell divides into two parts. Now they can divide either transversely or longitudinally or to an irregular manner. If they divide irregularly, like say for example in amoeba what it happens, it's called a irregular binary fission. In transverse fission they will divide along the horizontal axis. Now, like in Paramecium and Planaria, this is a Platyhelminthes, and Paramecium, you know, that's a protozoa. They transversely divide to form individuals. Like in Paramecium, there are two nucleus, the macronucleus and the micronucleus. So the macronucleus divides, and then the micronucleus also divides. Then the cytoplasm separates into two units. That is called transverse fission. In longitudinal fission, which takes place particularly in euglena, there is a vertical division along the blepharoplast, and then the flask-like structure, the mouth also divides into two parts, and longitudinally two flagellas are developed, and then they divide longitudinally. So that's called a longitudinal fission. In multiple fission, of course, it occurs in various organisms, like in Plasmodium, you know, like the malarial parasite, that during unfavorable times, suppose if in an individual, when the Plasmodium is hiding, suppose the individual is taken a medicine, then the Plasmodium goes to the liver or in the spleen, and then they form a three-layer cyst wall. Inside the cyst wall, the nucleus divides into multiple units having little bit of cytoplasm surrounding them. Now this cyst wall protects it from any type of unfavorable condition. And when inside this amoebulae, you know, or the or the spores, they remain till the unfavorable conditions are gone. And when the unfavorable conditions are gone, the cyst wall breaks, and these new units, they come out in the form of spores. Now there are the second type of asexual reproduction is budding, which occurs in yeast, where the protoplasm bulges out, and this protoplasm, you know, like bulges out from the single cell, forming an outgrowth. And like say for example, this is the nucleus, then from here the protoplasm will bulge out like this, the nucleus will divide, and then the nucleus separate into two parts, and again another will form like this. They go on increasing. That's called a budding. In algae, of course, Like in certain algae like Protosyphon, this type of budding is seen. In hydra and sponges, of course, they are multicellular. So there what occurs is that there is a small outgrowth on the multicellular organism like hydra or sponge. This outgrowth arises from the cylindron and they form a cylindric cavity with the tentacles. And if they are detached from the parent body, they give rise to new individual. So that is a type of budding which is seen in multicellular organisms like hydra and sponge. So we say there is an outgrowth from the body parts. Now the third type of reproduction is called gemule formation, which is found in certain freshwater and marine sponges, where inside the old parent body usually they form a cyst wall, a layer which may be internal thin membranous layer and a hardened outer chitinous layer with spicules. There is a small opening called the micropipe, and this type of gemules occurs inside the parent body. Now when the parent body dies, this gemule remains inside it, and then when the parent body dies, they are released in form of like spore-like structures, and they move out of the parent body and develop into new individuals like sponges. In marine and freshwater, there is slight difference. In marine, the thick chitinous layer is not there, only the membranous layer of these gemules are formed, but in freshwater both are formed. So they are hardened ball-like structures which are formed inside the body of the sponges. So gemules are formed in sponges with chitinous layer and their membranes, they are laden with food. They give rise to the new sponges. Then we have regeneration. If you cut a particular part of the hydra or echinoderms like starfish, there is an outgrowth or the body part regrows into a new individual. Like in hydra, if any hydra is cut out from longitudinally or transversely, it gives rise to a new hydra. In echinoderms, like say for example the starfishes, if the limbs are cut out, their new limbs grow. So that's regeneration. Fragmentation you find in certain types of algae like Eulothrix and Spirogyra. A small cut part of the Spirogyra can give rise to a new individual. And sporulations are also seen in certain small organisms. So till then, this is the part which we have covered today in today's topic, and this probably is the last class. If you have any question, you can go through this. You can read the chapter and you can see the video. If you have any related question, you can put up the question. Thank you very much.